Each cell in the body serves as the stage for an intricate ballet performance with thousands of dancing molecules. Each dancer needs to perform their part precisely in order to preserve the meaning of the dance and keep the cell alive. But inside the brain cells of people developing Alzheimer's disease, a dancer called RAP5 becomes hyperactive, running all around the stage and disrupting the other acts in the cellular ballet, which may contribute to cell death and dysfunction. RAP5 seems to be missing its cue to leave the stage. If we can understand what that cue is, we may have a better chance of successfully treating Alzheimer's disease. Finding this cue has been challenging in human neurons, but luckily it's much easier to identify the equivalent dance performed in budding yeast. And so the goal of my thesis was to describe this molecular dance, specifically the steps in between RAP5's entry and its exit from the stage. I was able to follow each dancer by putting tiny fluorescent protein tags on them and then looking at the cells with a microscope. I also made mutations to remove or alter one dancer at a time, and then measured how many mistakes other dancers made. We knew that our protagonist, the enzyme that turns off RAP5, needs a helper protein, but we found that this protein often seemed inactive. To find clues as to why, I screened over 4,000 yeast proteins to find others that may take part in this dance. First, I found two antagonist proteins, which seemed to restrain and disguise the helper protein, keeping it from performing its role, and therefore allowing RAP5 to remain active. I also identified a catalyst protein. My data suggests that this catalyst arrives and chemically modifies the helper, removing the disguise and allowing it to escape from the antagonist to finally signal to our protagonist that it's time to dismiss RAP5. There's still more work to do in understanding how this dance is connected to other scenes and acts in the production. For example, why is it so important to delay RAP5's cue? And what exactly determines the timing of the catalyst's arrival? Also, which details are the same in human neurons and which are the differences? But finding these proteins and describing their roles in this story effectively widens our spotlight focused on RAP5 and gives us more context to interpret other movements we might see by other dancers. This could ultimately help to explain why RAP5 is missing its cue in Alzheimer's disease, and could suggest ways that future therapeutics may be able to adjust the choreography to restore the intended storyline and improve